Hi, I'm Nicholas Roth, a computer engineering major at SJSU. As you may have guessed, HIV is likely to come up during this talk. HIV, the human immunodeficiency virus, attacks the immune system until it becomes severely deficient, which results in a condition called AIDS, the acquired immunodeficiency syndrome. As a side note, there are actually a lot of different types of HIV. We're going to focus on the most common and the most studied type, HIV-1, group M. If I were to get HIV from sex or sharing needles in the US, this is probably what I would get. Here, you can see the things we'll be going over today in this very brief talk. So, as you can see here, HIV-1 infection starts when one of the viral loops from the envelope attaches to the CD4 membrane protein of a CD4 positive T cell. Another loop attaches to a co-receptor, which I'll discuss later. Then the virus fuses its membrane, which has viral RNA and reverse transcriptase in it, with the target cell. This is mediated by envelope protein GP41 if anyone's interested in that. So I mentioned a co-receptor earlier. In R5 tropic HIV, the second loop attaches to CCR5. In X4 tropic HIV, it attaches to CXCR4. And in X4R5 tropic HIV, it can attach to either. The X4 and X4R5 tropic behaviors typically come from mutations after infection because they aren't really as virulent, and so they don't tend to be responsible for the initial infection. This makes CCR5 important in preventing infection and in curing early stage infections. So, once the virus is inside a cell, it uses this molecule on the left, a reverse transcriptase molecule, which it's a sort of DNA polymerase that bases the DNA output on RNA input. This is the computer engineer in me speaking. So HIV, being a retrovirus, all retroviruses do this. They come into the cell, they use reverse transcriptase to turn their RNA into DNA, and then they use an integrase, as you can see on the right, to embed the DNA in the host cell, and eventually it will be activated by any of a number of factors. So make sure that you use condoms and don't share needles, because once that happens, once HIV gets into your cells, you have a very nasty infection that's pretty much impossible to cure with current medical knowledge. If you do get HIV, you won't notice much more than annoying viral symptoms about 2 to 4 weeks after the infection. This can be disturbing, especially if you've been sexually active and get sick, since current tests aren't effective at detecting it until 6 months after infection. This is why it's important to get tested regularly, even if having safer sex, because accidents do happen. And of course, it's especially important if you've had unprotected sex or shared needles. Once the HIV in a cell becomes active, the host cell will start rapidly producing viral proteins. It will rapidly transcribe and translate those HIV proteins over and over again. And the protease will assemble them, and they will just start to bud out of the cellular membrane. This is bad for cells. It kills them, which brings us to the next point, AIDS. One important distinction between AIDS and HIV is that AIDS is a condition, while HIV is actually just a virus. Left untreated, an HIV infection will become the AIDS, the condition, once the CD4 positive T cells are gone or substantially decreased in number. These cells are important because they collect antigens, which are sort of like parts of the cellular membrane, cellular walls of various pathogens, and they deliver them to B cells, which take those antigens and they produce what we call antibodies that will seek those out and bind to them and prevent those pathogens from exhibiting their pathogenic behavior. An immune system with AIDS, though, is a lot like a prison with no guards. It's very deficient at its job. AIDS symptoms are generally related to difficulty fighting infections. I would suggest checking Planned Parenthood's website for a complete list, but symptoms of AIDS can include thrush, sore throat, bad yeast infections, chronic pelvic inflammatory disease, headaches, and rapid weight loss. And I just want to clarify that thrush is when a sort of whitish film forms on the tongue. Fortunately, there's been a lot of progress recently towards a cure for HIV. One path, highly unpopular for ethical reasons, involves using CRISPR-Cas9 to modify embryonic DNA. This is considered a germline edit because it's going to be passed on to the baby's offspring. This edit introduces a mutation called delta 32 into the CCR5 gene. We talked about this earlier. It's what most new HIV infections use to latch onto a cell and insert itself. And, well, this prevents that from working. The thing is that this isn't clearly necessary, and germline edits haven't really been explored, and they could be potentially pretty bad for humanity. The more promising approach involves using the same CRISPR-Cas9 system to modify immune stem cells extracted from a patient and reimplant them. A 2017 study, and you can see the results for this on the right, showed that this appears to work very well for lab mice. As you can see from the plot, the viral load substantially decreased by at least 2 orders of magnitude as measured by parts of viral RNA in the bloodstream of treated mice as compared to untreated mice. Another bonus of this approach is that, unlike the crazy scientists, this one is peer-reviewed. So the current treatment options for HIV are quite effective, albeit a bit less glamorous than a cure. They tend to revolve around something called ART, which stands for antiretroviral therapy. It's a group of medications that inhibit HIV, including fusion inhibitors, which prevent the envelope proteins GP120 and GP41, I think, from attaching to an immune cell and fusing with its membrane. reverse transcriptase inhibitors, which prevent the viral RNA from being turned into DNA once it gets into a cell, protease inhibitors, which keep the mechanisms that would normally put this virus together from working, integrase inhibitors, which prevent the retrovirus, HIV in this case, from integrating itself with the target cell's DNA, and CCR5 antagonists, which block the CCR5 receptor, so the virus can't grab onto it to enter a cell. One important thing to note that these are all ways of slowing the progress of an HIV infection, but they're not cures, so it's important to stay safe.